Biotechnology Day. So you guys get to help us test it out, figure it out, and hopefully we're going to be able to do this for more high school students in the future. Okay, so tools. Physical, chemical, organisms. You hear those words he threw out there? Okay, somebody else. Yeah.
The use of living organisms to create products to benefit people or things.
Okay.
My name is Kai. You guys can call me Kai. I have my PhD in computational biophysics. Does anyone know what that is?
Nope.
But what it means is that I grew up playing video games, and so I started programming in order to find the best cheats in video games to figure out how to win or how other people were winning. And then I figured out how to beat the cheats. But the human genome had just been sequenced. And lo and behold, it was really a lot of fun to hack into the human genome server and get a lot of information that I could use towards my project. Nonetheless, the skills that I had, which had nothing to do with science in general, helped me to become a better scientist. And how would you represent a network?
By letters.
Let's do an exercise. Someone give me their name.
D-A.
Maybe these lines are for you to draw. I can keep doing this, right?
Yeah.
And lo and behold, you see that?
I'm a box.
It's usually not what happens, right? Usually when someone is sick, you try to stay away from them because our intuition is there's danger there. Well, it turns out you guys are not very good at that. I don't know what that says about you. Either you like red dots or you like taking days off of school. I don't want to know the answer.
¿Por qué no los dos?
I lost myself.
I found you, I got you saved.
No, I was already saved. Is it the right campus?
Alright.
Maybe being
Chris, do
around
you want to
people.
tell people what we did?
We made the chance of getting infected 50%.
Okay, so you guys had half a chance to not get the disease, which was really sweet of her. Because, I would. This is kind of, in essence, what I do, except I do it with diseases and figure out which cells are most likely to be crippled by particular diseases, such as cancer and neurodegenerative diseases. And you do that by understanding the behavior of genes. So, how many of you guys think that you could be a computational biologist?
Welcome, everyone, to the lab. My name is Kristen Perkins. You can call me Kristen. So I'm going to tell you, we're going to do a lab today that is based on some work that scientists do. There are two tubes on your plate. One is a green liquid, one is a clear liquid. The first thing that I want you to do is you are going to just notice some properties about this liquid. You're just going to do a little bit of exploration. See what happens when you mix them together. When you mix them together, I don't want you to pour the tubes together. You're going to mix them together in a very purposeful way. So I want you to see what happens when you suck up some of the green liquid and put it into the clear liquid. That the green liquid that you're using was actually a biomaterial. What does that mean? What does bio mean? Just that word from life. Okay. So if it's a life material, biomaterial, if it's a living material, that might mean that it comes from a living organism. Good, a living organism. So the green goo is actually called sodium alginate. From algae. So it actually comes from brown algae. We color it green with food coloring. That's all that is. But it starts out as a brown powder. We mix it together with water. And this biomaterial can be used to form a protective shell, like a protective layer around cells. We want to cryopreserve them because we need them to stay alive for a long time, for years, outside of a body. Cryopreservation. Heard that before? Freezing it. Maybe the alginate can protect the cells, and then you can cryopreserve them. So then the scientists said, well, how are we going to get the cells inside of the alginate? Or how are we going to make the alginate B form around the cells? How can we do that? So you answered one of the first questions that the scientists had, which is how do we use this alginate? Can we use it to make beads? Okay, figured out some ways to do that. Well, now let's figure out if we can make the alginate bead with cells inside. Okay, so hold it and roll it. That works. Okay. See people mixing it with calcium, but now there's an entire field of science called oncofertility. Does anybody know what the word part onco means? Like an oncologist or oncology? Yeah, cancer. The chemotherapy, the radiation, is going to damage her fertility. More than likely, she will, if she survives, when she survives the cancer, when she's better, down the road, if she wants to have children, she can't do that. And so they wanted to be able to take out the ovary, take the egg cells, and be able to cryopreserve them. So that someday, down the line, when she's healthy and when she's ready to start a family, that those egg cells are available. And scientists are doing this.
Hi everybody, I'm Darcy Patterson. I did my undergraduate education at Hawaii Pacific University.
Hi, I'm Gia Wu. I'm from California. I did my undergrad at University of California, San Diego.
Hi everyone, my name is Biotechnology. I grew up in Florida and then went to Boston. Went to MIT for my undergraduate and studied materials science.
Jeff Liu. I'm a PhD student in the IHSCA program. I grew up in the San Francisco Bay Area, and I did my BA in molecular cell biology at the University of California, Berkeley.
This is Justin Finkel. I grew up in upstate New York, and I did my undergraduate in bioengineering at Cornell University.
If you think about the introductory textbook, one sentence that they put into a section about the nucleus, that took 30, 40 years of just work, and just it's a lot more complex than you can ever just get from a textbook.
So athletes train, and they're focused all the time on making their toolkit, making their muscles, making their body stronger. We have to do that really with our minds. So we're constantly reading, we're constantly learning more about our field and other fields so that we can really apply that research.
What I do is just look at kind of what Jeff was saying, looking at the bigger picture. So I've heard this before that getting your PhD is like making some small niche improvement in some category that maybe no one will ever read the papers they publish. But I really value what I'm doing, and I think that's what's most important.
I didn't know what I was interested in when I was applying to graduate programs originally, and I didn't know So I wasn't able to focus, and I actually applied my first year at undergrad to graduate program, and I got rejected by all of them because I wasn't asking the right questions. So most of the time we're thinking that we'll have a question to ask and we'll have a straightforward answer and we'll get there, no problem. But if you haven't phrased your question properly, if you don't know what you're interested in, if you don't know what problem you want to solve, it's really hard to get there.
So I actually was an NCAA athlete for all 4 years of undergrad, and that was a huge time suck. It was like, yeah, practice every day, 3 hours a day, plus practice on Saturdays, it was 4 hours. I was on the fencing team. So kind of unique. It's like team captains. Actually, it's always good to have a balance between work life and personal life.
So I think it's really also very important to have things outside of research that make you happy. So I've always been a dancer, so I'm a dancer at Western and Western undergrad. And I also box, so I like to do things that are outside of the lab to like let go of my aggression sometimes. I also travel a lot, cooking and everything. I think there's just like, no matter what you do, whether it's research or not, just find a balance of things that you really love and work and
you should just have sort of like set goals and not just like, to get an A on this test or an A on this grade, but sort of approach it as like, these are the things I need to do to reach my goal of where I want to be in the future, and like not just approach it sort of like drudgery, like I have to do this, but like these are just like the skills and the things that you have to pick up. And so really being motivated by that fact of like, just that daily self-improvement, getting better at one thing every day, or learning something new every day. I think that's a really important thing to keep in the technical life.
In STEM fields, if you're really interested in being an engineer or a doctor, it's really easy to label yourself. You're going to take all these classes that are really hard. I was in a class of 300 very often, and oftentimes the average for a test was 60%, so that's like a D, right? And there are always people that will have set the bar. But if you label yourself as, oh, I'm not very good at physics, or I'm not very good at organic chemistry, that shouldn't be your focus. It should be more about whether or not you like solving problems. And you have to push through and learn the material. Ultimately, we're all learning the same material. Some of us are learning at different paces, and it's okay. As long as you like solving problems, you can push through whatever courses are really happening.
If you're happy at the end of the day, then there's nothing, we don't do it because we think we're going to win a Nobel Prize. I personally do it because I think it's really interesting stuff.